Thank you all for being here to listen to me again, talking about defense and the program we're doing in immuno-oncology. So what I thought of doing this time is to give you again an overview of all the technologies, or not the technologies per se, but the different applications where the technology has been applied, just to remind you a little bit about the different programs. And hopefully in the next IIF meeting, I'll be talking about new developments that are basically going on at the moment. So again, what is the mandate of defense therapeutics? As you know, if you have, for example, an antibody or mRNA molecule, any kind of biomedicine that you'd like to introduce into a target cell, these molecules or this biomedicine will be taken up by a process called endocytosis, which will lead eventually to the development of an endosome. So these structures are usually there to make sure that whatever protein or biomolecule that has been captured could be degraded into smaller particles or smaller portions that will eventually lead to the cytosol to a certain point. And it really makes sure that this happens by going through what we call a maturation process, where you've got high acidity going on, the production or the activation of proteases that will start chopping off different parts of the molecule. You'll have reactive oxygen species that will cause damages. And eventually, to really complete the destruction of the molecule, whatever has been captured, these endosomes will fuse with the lysosome to really destroy everything to small particles. And so if you think about it, this becomes a major hurdle for any kind of technology you're trying to develop or product you're trying to deliver to the cell, whether it's a protein, RNA, DNA vaccine, antibody-drug conjugates, CRISPR, and whatnot. So the way that defense has really worked on to be able to bypass this limitation is to develop and apply the accum technology. So as you can see on this panel here on the left side of the slide, what the accum technology will do eventually is that it will cause endosomal break, or it will destroy the membrane of the endosomes, allowing these captured molecules to diffuse out of the endosome into the cytosol. So it's an endosome to cytosol escape. And if you can think about it, this application could be directly useful for any protein or even cell-based vaccines. You can think about the mRNA vaccination. You can think about, enhancing the accumulation of INR2 or Kadcyla for breast cancer, or rituximab for hematological malignancies. And what's really beautiful about the accum technology, which, as you can see here on the top, the accum molecule is composed of two components, a bile acid and a nuclear localization signal or peptide. And so you can play with these two components to generate libraries of molecules. And the good thing about this is that you can end up having novel molecules exhibiting unexpected pharmacological properties. And that's really, that led us to one of our programs, the AccuTOX, where now we discovered that we can actually tweak the molecule and design small anti-cancer molecules that are really potent at inducing cell death. So how does the accum technology work visually? It works visually. So if we, for example, think about a cell that where you could actually overexpress a fusion protein, the GFP fused to the human galectin-3, the importance of this molecule is that the human galectin-3 will only bind to carbohydrates that are usually found on the extracellular part of the cell, so on the outside part of the cell, or within the endosomal lumen, which is really just a structure that is formed by really taking parts of the extracellular membrane and really creating this kind of an endosome. So really, if you express this fusion protein, all what you're going to see is green fluorescent protein all over the cell. Now, if you add an antigen, nothing's going to happen. However, if you have an antigen that is linked to the accum is then going to break down these endosomal membranes, allowing this green fluorescent protein fused to the human galectin-3 to diffuse within the endosomal lumen and thus interact with the carbohydrates in the inside of these structures. So this is a great example showing you visually that the accum technology is really able to allow proteins to move in and out of the endosome. In this case, going inside the endosome because the human galectin-3 has to bind to carbohydrates, but in reality, whatever is entrapped in there will eventually be able to diffuse back into the cytosol. So of course, we've applied this technology in different verticals, one of which was to demonstrate that we can enhance significantly the activation of T cells by re-engineering dendritic cell vaccines, which have been around for over 30 years now in the field of cancer immunotherapy. We've demonstrated that the accum technology could enhance by at least 50 to 100-fold the activation of T cells. And if you're really interested to know more about the accum technology and its applications, I strongly encourage you to go read a review article we published in the journal Molecules that really explains more in detail at the scientific level how the accum works and how it could be applied to different strategies. So this is now the where we are in terms of our developmental pipeline. So we've got really a cell vaccine that is moving very well. We call it the ARM, and I'll be giving you an example of each one of these different. I'll be talking briefly about each one of these different products. So the ARM vaccine is a mesenchymal stromal cell vaccine that has been actually reprogrammed with an accum variant to behave as an antigen presenting cell. And right now we're at the level of CMC, and we're hoping to be able to actually enter a phase 1 trial by the end of this year. We also have the AccuTOX, which is the small molecule that we spoke about that is able to actually exhibit anti-cancer properties, which we're also trying to push right now into phase 1 trial. It's at the CMC level. We're working with City of Hope in California to be able to initiate a phase 1 trial against melanoma. But we also were able to initiate another formulation to be used directly to treat lung cancer. So it's an intranasal formulation. In terms of protein vaccination, we finished all the preclinical work related to a protein-based anti-cancer vaccine targeting cervical cancer. We're just in the process right now of securing a CMC site to be able to move on again to phase 1. And of course, we have the Antibody-Drug Conjugate program, as well as the mRNA vaccination program, which we believe will have a great impact because we can apply it to basically any mRNA vaccine in development, whether it's in collaboration with current companies or through the development of our own mRNA vaccine. So I'll give you again just brief examples of how these different technologies work. So for the ARM vaccine, which stands for the A1 reprogrammed mesenchymal stromal cell vaccine, this is a program that is designed for solid tumors. So the idea is to target, in this case, melanoma as a start and eventually move on to other indications. So again, the big question is, why mesenchymal stromal cells? What makes them special compared to other antigen-presenting cells or even dendritic cells? Well, first of all, MSCs are very easily harvested. So you can take a single small bone marrow aspirate from any individual, and you can easily put these cells in culture and expand them to billions of cells, which you can freeze and thaw. So it's you can end up having high yield, simple expansion approach, cost effective. And of course, you have a very good safety profile. So there has been more than hundreds of clinical trials conducted with MSCs, and none of them have shown any negative impact on patients' health. What's really interesting also is the plasticity of these cells. So MSCs could be actually oriented the way you want them. If you find the right conditions, you can reprogram them to deliver something of interest. So in this case, what we thought, in the past, I'm highly interested in these cells for the last 10 years. So I was able to engineer them to make them antigen-presenting cells by overexpressing the immunoproteasome complex, the thymoproteasome complex, and we've shown that in both cell products, these vaccines were able to control, if not eradicate, established tumors. We've also shown that we can pharmacologically manipulate or reprogram these cells. So there is no genetic engineering, there is no insertion of genetic material. It's just a pharmacological agent you can add on these MSCs, and then you can reprogram them to become antigen-presenting cells. So this approach is kind of interesting to us because it really simplifies the manufacturing process as opposed to really generate an lentiviral vector under GMP conditions and move forward. So based on that, we conducted high throughput screening, a very small high throughput screening with different accum variants, but we've tested two approaches. The first one was using accum-linked antigens versus admixing the accum variants with the antigen. And what we noticed in this case is that if you basically use the OVA-linked accum variants, and these are the different variants we've tested, we were not able to actually have any kind of an effect on the cell. So the cell was not able to cross-present antigen. And by cross-presenting, I mean you capture an antigen, you process it, you present it on the cell surface to activate T cells. The interesting part, however, is that if you admix, so you just mix the A1 variants with the OVA protein, We had one hit we called A1, and that hit was actually able to stimulate T cell activation. So this is a very interesting approach that we thought, could be very simple to translate into clinical trials, because then you can just expand your MSCs, add your antigen of interest mixed with A1, and there you go, you've got an antigen presenting cell. Of course, we started wondering what could happen in these cells. Why is the A1 reprogramming them, or by which mechanism is it programming them? And what we found is that there are a variety of different involvements of Frost production, metabolic reactivations, and whatnot. But what's really interesting in this case was the activation of the so-called unfolded protein response. So this is a response that happens in a cell, usually when you have, for example, some sort of a protein-based stress. So aggregates of proteins accumulate within the cell. The cell is going to find ways to activate different proteins like heat shock factors or proteins that will then go and try to unwind these complexes or aggregates to make sure that the cell doesn't actually doesn't die. But the good thing is, in our approach, even though we have the unfolded protein response activated, the cells don't die, meaning that the protein aggregation combined with the A1 is really helping drive the antigen cross-presentation process in these cells. So, of course, I'm not going to go over all the different in vitro studies we've conducted. We've got tons of them, and we were hoping to be able to publish all of these in a couple of months from now. But the big question is, can we use this vaccine to treat established tumors? And the answer is yes. So if you take, for example, an allogeneic vaccine, so no compatibility with the recipient, off-the-shelf allogeneic product, you inject it to animals with pre-established T-cell lymphoma. So this is a solid tumor subcutaneously in these animals, and these are immunocompetent animals, by the way. So you transplant the tumor, you give the vaccine, so 2 dosing, along with the immune checkpoint anti-PD-1. If you look at the tumor growth over time, you can appreciate that the combo group, so the arm vaccine with the anti-PD-1, actually cures 90% of these animals. So the tumor actually disappears, and it doesn't actually grow. In fact, what I want to bring to your attention on top of that is the fact that the vaccine on itself as a monotherapy was also able to actually control tumor growth. So this is something we don't really see in the DC vaccine world, because DC vaccines on their own, or when combined with the immune checkpoint blocker PD-1, will lead to barely a tumor delay or survival delay, or maybe 30%, 20 to 30% survival. So this is really outstanding as a piece of information. And of course, this is using the OVA as an experimental antigen, but we have reproduced similar data with the B16 or the melanoma tumor lysate, which is the model we're going to be using for our phase 1 trial. So the take-home message in terms of the suggested mode of action for the A1 is that once you have the A1 mixed with the antigen, it kind of forms aggregates, and we've got some evidence of that. These protein aggregates will then accumulate within the endosome. We have evidence that the A1 triggers through the NOx, the NADPH oxidase, or the NOx production of reactive oxygen species, which will lead to lipid peroxidation and break within the endosomal membrane, allowing this aggregate to be released in the cytosol and then directly being targeted to the proteasome for degradation and eventually presentation on the cell surface for T cells to see the antigen and be activated. So right now, we're moving very well in terms of developing our phase 1 trial. So we're starting the manufacturing at McGill in a cell processing center. We would like eventually to go for a 3+3 design for our phase 1, identify a dose that is actually well tolerated by patients, and eventually move on to a combo with an anti-PD-1 to see eventually if we can even see some therapeutic effect in these patients. Now I'm going to switch gears and talk about another application of the accum, which is really targeting, for example, cervical cancer, but using protein. Now we've discussed, the application in terms of the cell. What about accum in terms of protein vaccination? So why cervical cancer? Because we know that cervical cancer is caused by a viral particle, by the HPV. And we know so far that Gardasil 9 is doing a very good job in controlling the prevalence of cervical cancer. But there are 2 main problems with this vaccine. First of all, Gardasil 9 covers 9 different subtypes of the HPV. So you're protected against 9 different subtypes. So there are new subtypes that are basically, or new strains that are basically arising in Asia, for example, in China, that might be leading the next wave of cervical cancer over time. The second problem we're having with Gardasil 9 is it's only used prophylactically. So you can only give it prior to the infection, making sure that the patient doesn't have cervical cancer. So if the patient has cervical cancer, then he has a problem because the vaccine is no longer effective. So in our case, what we have decided to do is to basically look at the major proteins that are driving immortalization or transformation of the epithelial cells. And we decided to focus on the E7 protein because that's a central protein that drives the inhibition process of apoptosis. So it really blocks all the machinery, responsible for blocking growth, proliferation of cells, and eventually down the line, apoptosis. So by attaching accum to this E7 protein, we demonstrated through a prophylactic vaccination study that, in fact, our accum E7 vaccine will lead to 100% protection. And that despite two additional challenges that were conducted in these animals. Not only one single challenge, but we did two extra ones. And nevertheless, these animals remain tumor-free, meaning that we were able to generate a very powerful anti-tumoral response, or a very powerful memory response controlling these tumors. Not only that, but when we took that vaccine and we applied it now in the therapeutic vaccination setting, in other words, we transplant the tumor first and then we vaccinate. And here we did a vaccination along with different immune checkpoint blockers, just to show you that there is some synergistic effect with different immune checkpoints. You can see here the vaccine on its own is leading to some sort of a delay. But if you combine it with the anti-PD-1, the CTLA-4, and now we have data with CD47, you can really control tumor growth and really make sure that these animals are surviving longer and longer. And we're hoping to be able to optimize this response to really end up having complete regression over time. So one thing I'd like to remind you is that our vaccines are all synergizing with the different immune checkpoints, including those that are currently in development, such as LAG3 and TIGIT, that are basically making their way to through the clinic. So we're also looking here we're discussion right now to move on this program to phase 1. So we have different sites that we're actually talking to be able to identify the PI to be running the phase 1 trial sometimes again this year. Now, I'm going to show you another example of how you can apply the accum, and that's through the AccuTOX program. So initially, as I mentioned to you, you can take the accum, link it to an antigen, add it on DCs, and therefore you have an activated dendritic cell that will be able to drive T cell activation. What we thought initially of doing is to simplify the manufacturing. We said, okay, well, can we just mix accum with an antigen added on DCs? And we ended up having cell death, which drove our attention that when the accum molecule is freely available, it is able to kill cells. And so we started wondering whether this is actually true if you apply it to different cancer cell lines, so the T cell lymphoma, colon cancer, melanoma, and breast cancer. And as you can see, the molecule can really drive apoptosis in all of these different tumor cell lines, but at different rates. So for example, it was weaker in melanoma. So by starting to work with the different variants and modifying the bile and the nuclear localization peptide, we ended up identifying AccuTOX 001, which is a variant of the original accum molecule. But as you can see, even though we lowered the dose significantly, we were still able to kill 100% all of these tumor cell lines. Not only that, but we tried, I think, over 11 different cell lines, including human and murine, and they were really all eradicated by this molecule. So with these data in hand, we moved ahead and we applied it in combination again with anti-PD-1, anti-CTLA-4, anti-CD47, which are 3 different immune checkpoints. Here, the drug was delivered intratumorally, so the tumor was established, and this is a T-cell lymphoma model. We've done it with breast cancer and melanoma. You can insert it directly, inject it directly in the tumor, and give the immune checkpoint blockers. And as you can see, the CD47 as well as CTLA-4 were doing very well in controlling the tumor, and the animals remained alive up to day 40. So we always stop our experiments at day 40 because we want to keep it consistent, but these animals actually survived much longer than a month and a half. So again, showing that the AccuTOX could synergize very easily with different immune checkpoints. So I'm pleased to announce that, in fact, we are working with City of Hope in LA in the US to be able to really push that AccuTOX into phase 1 in melanoma patients. So we're hoping to have IND filing by the summer and eventually move on with the Center for the Treatment of Solid Tumors, specifically melanoma in this case. But having said that, the AccuTOX attracted a lot of attention because this molecule seems to be very potent at driving cell death or apoptosis. So then we asked ourselves, can we start off playing with this formulation and develop something that could be given intranasally, so non-invasive approach? And the first thing we thought of was lung cancer. So why? Because this is a commonly spread cancer. It's able to metastasize and cause death of the patients. Usually the treatment types are surgery, chemotherapy, radiotherapy, some form of immunotherapies, but unfortunately the survival rate of these patients is quite low. So we're talking about 18% for a 5-year survival rate in lung cancer compared to other cancers for which similar therapies are being used. So based on that, we said to ourselves, is there a way so we can apply AccuTOX for lung cancer? And therefore, what we did in this case is we conducted a single administration intranasally to identify the maximum tolerated dose. So we tested 2 different volumes, one volume that will remain in the intranasal cavity and a larger volume which will make it to the lungs. And as you can see, we identified 5 milligram per kilo as the maximum tolerated dose. Now, when we did repetitive dosing using, again, the same volumes, we found that, in fact, you have to lower the dose a little bit to 3 milligram per kilo. Otherwise, the animals will start suffering or having some sort of side effects, which means that our maximum tolerated dose was 3 milligram per kilo. So, and here repetitive dosing, we're giving 6 administrations over 2 weeks. So having done that, we then moved on and delivered the AccuTOX intranasally into animals with pre-established lung cancer, and we delivered the immune checkpoint PD-1 along. And as you can see, this is a pictogram of the lungs. The black dots really represent the tumor nodules. And as you can see, if you deliver the AccuTOX with the PD-1, you get a pretty dramatic decrease in the number of nodules in these lung cancers. So this is a fantastic piece of data because it shows that the AccuTOX could also be applied or could also be formulated to be delivered to the lung and eventually fight lung cancer. And right now we're working with a U.S. company to be able to actually use or deliver the AccuTOX using a spray device. So now I'm going to switch to the antibody-drug conjugate program. So as you know, ADCs are composed of 3 components: antibody, the linker, and the drugs. And of course, the purpose of the ADC is to be able to bring to a target cancer cell a payload, which is a chemotherapeutic agent. Now, we applied the accum on Kadcyla, which is one of the most common breast cancer ADC. And as you can see, using 2 different cell lines here, we were able to show that if you link accum to the Kadcyla, you'll be able to really shift the sigmoidal curve to the left, meaning that now the antibody is way more potent and is killing more efficiently those 2 different tumor cell lines in vitro. We applied it to another antibody using the trastuzumab platform, and we were able to get something very similar in the sense that we enhanced by 70-fold. And here we're talking really different antibodies, so Kadcyla 10 to 100-fold, and this trastuzumab-based antibody by up to 70-fold. So now we know that accum could also enhance significantly in a remarkable manner the potency of ADCs. And right now we're conducting xenograft models, testing the potency of these therapeutic antibodies in xenograft models, breast cancer, to demonstrate that they're actually leading to a very powerful effect in vivo. Now, the last program I'll be talking about is how we can apply the accum technology on mRNA vaccination. So as we all know, mRNA vaccines have made it to the news because of the COVID. Fantastic work. Moderna BioNTech now are pushing their mRNA vaccines for cancer, coding tumor-specific antigens. But we're still facing the same problem, in the sense that this is an mRNA molecule. It's actually more sensitive than DNA or protein. And so you can imagine that mRNA needs eventually to reach the cytosol to be able to translate into a protein, and that protein will then be excreted outside of the cell to be captured and then mount an immune response. But if your mRNA is trapped within the endosome, you have the same problem. So in order to really bypass this problem, we're now working on attaching the accum on the mRNA and testing whether we can enhance significantly the therapeutic response to the mRNA. So right now, we've completed some initial studies with the naked mRNA, so just to show that we're able to mount an immune response. And as you can see, if you inject the naked mRNA, the immune response you can generate over 6 weeks is very weak. Now, if you give an adjuvant, you can stimulate it a little bit. But with these basal or basic results in hand, we're hoping that in the next month, when we're going to be receiving the OVA mRNA with the accum attached to it, to be able to enhance by at least 100 or 1,000-fold the immune response generated. So just to show you that this is work ongoing, and we're hoping in our next presentation to be able to present some very powerful data about how to apply the accum on mRNA vaccination. So overall, this slide here shows you that the accum technology is highly versatile. We've shown you that we can apply it to design cellular vaccines. We can apply it to protein or even mRNA-based vaccines. We can attach it to antibody-drug conjugates. If you have an ADC that, for example, needs to accumulate much better in the cell to be able to raise its therapeutic index, we can work with you to enhance significantly the potency. But we also have shown that we can work or play with the molecule to drive the development of novel small molecules with anti-cancer properties, which is really what's driving now precision medicine 2.0, as they say it, as they call it right now, which is the development of small molecules that have better therapeutic effect on cancer cells. So, of course, any of these technologies are highly synergistic with immune checkpoint blockers. So feel free to communicate with us if you have any questions, and I'll be more than happy to answer any if you have right now. Thank you.